teaches at UC Berkeley, the Department of Chemical and Biomedical Engineering, and she's a member of the Berkeley Synthetic Biology Institute and of the Synthetic Biology Engineering Research Center, which eventually I'll ask you to say 2 words about them. But anyway.
Okay. All right. Tell me when that's ready. Okay, so I'm going to talk today about something called synthetic biology. How many of you have heard the term? Okay, actually pretty good traction, which is great. What that really means is that we engineer biology, but we're doing it in a more methodical way than what has been traditionally being since, say, the 1900s, traditionally been done. But you'll hopefully get a much better feel for it with this talk. And I will try to focus more on the ideas of synthetic biology than all of the details of the method. So the idea behind the field and the reason that it's becoming a phrase is probably the most important take-home message of this talk. And what I first want to convey is that it has a lot of potential. So when you look at emerging technologies, synthetic biology on the World Economic Forum's top 10 was number 2. But actually, all of those highlighted in red on this list could be impacted or directly affected by advances in engineering biology as well. So if we want to, for instance, find new and improved ways to make food from the same amount of soil, louder? Okay. From the same amount of soil, make more food for people, that at some point will probably require figuring out a better way to manipulate organisms. If we want to have more personalized medicine, in other words, when you are diagnosed with cancer, you get a treatment very specific to your type of cancer within the context of your body, that again could make use of some of the methods that we're developing in synthetic biology. So even though it's number 2 on this list, it's also kind of inherent in all these others, which makes it quite important as an emerging technology. Okay. So why is it an emerging technology? Why is engineering biology suddenly something that is new? When in theory, even in the 70s, we had recombinant DNA. What's so different now? And the answer is represented on these 2 graphs. I hate to put graphs in, so I'm going to try to explain these pretty carefully because I think they're really important pieces of information. So on the left, you see the ability to both read and write DNA sequences. Right, so to make DNA sequences, so we can make the DNA that encodes all sorts of things in life in strings, at least not in chromosome form, but in long strings of base pairs. So that's the yellow. We can also read the sequences, so interpreting your own genome, getting the actual sequence in your body. We can do that. That's the graph that's shown, or the line that's shown in red. And you can see that for both of them with time, we're going, and this scale here is actually log scale on the y-axis. So we're going at the same pace as Moore's law. So most people are familiar with Moore's law, how many transistors you can fit on a chip. And that's really quick. Like, technology is advancing in terms of computing power very quickly. And our ability both to read and write DNA is going at that pace or even faster. So that's a tremendous increase in technology at our fingertips, and now we need to know what to do with it. And so just to put it in another way, in case that doesn't convince you all the way, this is the cost to sequence a single person's human genome. The cost a little over a decade ago was somewhere on the order of $100 million. Right? It costs a lot of money. And now you can probably, this is actually a little bit dated, so 2011, I'd say we're down under $10,000 per genome. So that's a lot of information. That's a lot of DNA that we're getting the sequence of in each person. And we can do that, and it's getting faster and cheaper. Still, it hasn't slowed down at all. In fact, it's accelerated, if anything. So we have all this information about what's encoding all of our organisms, and we have this ability to make a lot of sequences. And the question is, what do we do with that information, and what do we make, and how do we make it? These are really important questions. So is a cartoon kind of depicting a video game version, so very artificial. But the idea is that we can recode DNA now to create a new function in an organism. We're not great at this yet, but we can do it. And we can especially do things like make a new function where it used to make ethanol. Maybe we can make an organism, instead of making ethanol, make butanol, which would be a better fuel than ethanol. So we can make incremental changes at least. And doing this is a lot like rewriting some computer code to tweak something, right? So getting an entirely different function right now is a lot less feasible, but that's where the field would be going. The biggest application space right now for synthetic biology is actually in the production of fuels and chemicals, which is why I used that example a moment ago. We can make, very well, we can make medicines in organisms, right? A lot of small molecule drugs. So in other words, something like the anti-malaria drug artemisinin, which is now, I think, to the point of actually being distributed in Africa at a very low cost, is being produced in yeast. The many, if not all, protein therapeutics that I know of are synthesized in an organism. And so that includes things like Herceptin or Avastin. So there are a lot of drugs that you probably have heard of that are made out of protein, and the protein is in turn made in an organism. And to get organisms to make these things is not usually natural. We usually find a natural source, but we can't scale that up. We can't make a lot of it in that natural host, so we have to move it to something new, and that's what synthetic biology does. Lately, let's say the past 10 years or so, it's been especially important to a lot of synthetic biologists to make an impact on the world that's helpful, and so particularly with respect to the environment, there's been tremendous investment of time and resources into making biofuels. So these are fuels that have been generated, so in some way sunlight is converted into a form of energy that's stored in plants, so biomass, or algae, something that's growing, and we can convert that into a form that we can use, so a liquid fuel for jets or for cars. So biofuels, and at the same time biochemicals, because there are a lot of things we get from petroleum that do not include fuels, but that are chemical precursors. So probably your baby's diapers would be made of polymers that are actually made from derivatives of petroleum. And we could replace all of that carbon usage with something from a renewable source if we can just figure out how to make the molecules in organisms. And so we're figuring out how to do that. And then finally, even the precursors to a lot of fabrics. And by fabrics, I mean carpet fibers or particular blends of cotton-like material that form some of the textiles that now, at least in the couture lines, are being incorporated from organisms that have been engineered to produce them. So it's all around us. We don't really think about it that much. What we can do with organisms, with living things, to help them produce exactly what we want, so we don't have to manipulate it so much. So we have this explosion of technology, right? We can read and write DNA sequences. We can recode that DNA for new functions, albeit incrementally different in most cases, and then use them to synthesize chemicals, drugs, and fuels from renewable sources. That's not the main idea, I think. The really key point is where we're going to go with this. Where else can we go? This is what we've been doing. In fact, we've done a lot of these things from the mid-'80s, I'd say. So of course we're going to be able to make more fuels and chemicals. This is what I see right now in the synthetic biology community. A lot of people are coming up with really clever ways to make a variety of different fuels and chemicals from a renewable source, say grass clippings or grasses that grow in marginal land. So that's one. But I also see a lot of work in several other areas that I'm going to highlight. So there's this idea of bioelectronics, interfacing biology with inorganic, so computers. So imagine a wire that's somehow connected to a living organism. And either sending a signal down in digital form, because that's usually how the signals are going to be transmitted, and telling the cell to do something either with the on or the off portion. And then the cell has some output depending on what the wire is saying. Or vice versa, maybe a bacteria that's connected to a wire actually depositing electrons. And so you have a living battery, you have some organism that takes in sunlight and directly converts that into electrons that you can use out in the field, maybe where there isn't any other power. So this would be directly using the organism for energy rather than going through an intermediate step where you store it and then you have to break it apart and make a new molecule. So bioelectronics. Biomaterials, I've talked a little bit already about how we can make some of the precursors for things like carpet fibers and certain textiles. There are a lot of materials out there that are entirely produced in organisms already, right? Cotton is one of those that actually already comes, so we just kind of refine that. Spider silk turns out to be a really interesting material as well. It's stronger than steel, tougher than Teflon or Kevlar, depending on what you want to call it, and very elastic, so you wouldn't want to use it as a bulletproof vest. The bullet would be stopped, but not before it actually went in you with the spider silk fabric, so you can pop it back out, but that wouldn't be so great. But you can imagine that might have some utility in the world, to have these different materials with such different properties than what we have. And these types of materials turn out to also be made out of protein, which means that we have to produce them biologically. There's not even another way to synthesize them. And so we have to figure out how to get them out of things other than spiders. I don't know if you've ever tried to grow more than one spider at a time. It turns out they're pretty territorial. You can't scale that up. You can't make a factory with a ton of spiders inside and pull their silk. That just doesn't work. And so we need to find some other organism that can make spider silk for us that's more amenable to being grown in small batches that produce a lot. So more far out there ideas that are probably coming in a couple more decades, but the ideas are already here. The idea of altered environment. There was a Kickstarter a year and a half, 2 years ago now, I think, for glowing plants. And the idea is, behind this project, to plant a tree that has something encoded in it that makes it glow at night. So you don't need street lamps anymore. You don't need electricity. You just have the tree planted. It's doing its thing during the day. It's great for the environment. And then at night, it also glows and lights the path. It's a really cool idea. It's feasible to make a plant glow, probably not bright enough to be a street lamp, but the idea is already there, right? So we just have to do it. Of course, that's not natural. Plants don't naturally glow, but there's the firefly enzyme could be moved into plants. So altering our environment, that's one idea. Other ideas I've heard are making a tree, so engineering it a little bit, so instead of growing straight up, it actually grows into the shape of whatever it is that you want to build. Can we start to control the shape of the tree? So, you know, maybe it grows into the shape of a house instead of just straight up and down. Cutting out some steps. Again, we don't have the capability to do that yet, but people are thinking about what would it take to be able to do that? What in the DNA tells the tree to grow in particular shapes with the particular types of branches they have? These are all really interesting questions that come out of just trying to think about engineering these functions. And then finally, I already talked a little bit about personalized medicine. We have to learn a lot more about what all of the DNA means. So we can sequence the genome, but we don't actually know what all of those pieces mean. And so figuring out what they mean and how that's going to impact how every single drug is metabolized within somebody, how much more likely you are to get cancer than someone else to begin with. All of these things are questions that we need to answer still. And last but not least, I think the most transformative technology that's going to come from synthetic biology is probably not any of the other categories. It's going to be something completely new that somebody is thinking of right now, developing right now, or maybe they're in classes right now just learning about synthetic biology, but they're going to come up with something that can truly change the world, and hopefully for the better. So what is our challenges? It would be incomplete if I told you all of these possibilities without telling you what the problems is. And it turns out there are a lot of problems. And all of them pretty much have to do with the fact that biology is really complex. There's a lot of DNA in a human, for instance, right? And all these all these functions that it encodes interact in ways that we don't completely understand, and we need to understand that if we're going to, one, manipulate it, or two, have a chance at doing so in a way that's actually towards the goal that we want. So one of the ways that biology is complex, I'm just going to highlight a couple there. There are so many, I just want to give you an idea, but one of the ways biology is complex is that organisms respond to changes in their environment. So it's not like a test tube. So I think a lot of people that engineer biology like to think of it as an A goes to B reaction, but as soon as you add A, the situation has changed for the organism, and so it won't necessarily just go to B. It might go to B under one particular circumstance, but then on a different day it doesn't, because maybe the pH is slightly different, or the sunlight is at a different level, right? So all of these factors from the environment actually change the response that we get. Within the cell, things can change a lot. There's simple systems where you have some sort of input leads to some sort of output, and that's directly regulated by one component of the cell. That we can figure out, and that's the way it works, right? Because we can eliminate that component and we eliminate our output. But often in cells, they're regulated by something much more complex, where maybe there's 3 different components that are sensing the same input, and the way they react to that input dictates the output of the response. That's actually the way the human nose works, right? We don't actually have a sensor in our nose for every single chemical or molecule out there. We have just a few, and it's the pattern that your nose nerves actually detect. It's interpreted by your brain to be a particular smell, right? So the same is true for most of what's going on in cells. And it's great right now. We are really adept at working with simple systems, and we're just starting to learn how to work with the complex systems. Another way it's really complicated is that we need to develop new ways to organize activities inside cells. So right now, a lot of engineering biology resorts to putting new pieces of DNA in and treating a bacteria, so just a microscopic organism, as a sac for holding that DNA and turning it into enzymes. So it's just sacs, little factories producing what we want from A to B. But in fact, in nature, that's not how it works. There's usually scaffolds for all the pieces to make component A turn into component B. And so we're going to need to learn how to organize all of the activities that we want to put into cells, or else it's never going to produce all that we can dream of. There's 2 ways of doing the scaffolds. One is to kind of line up like an assembly line. Another is to compartmentalize. And I actually work in my lab. I won't talk too much about my own work today, but we spend a lot of time trying to figure out how to introduce new compartments, entire compartments into cells to house the artificial reactions that we're introducing. And so that's kind of inside a cell, how you might need to organize, but you also need to organize cells within a network of other organisms, right? Usually things are not isolated in a test tube. In fact, this right here is depicting E. coli that, so the center spot was placed on a petri dish and they were allowed to grow outward. The center spot is just well mixed with E. coli that either had a green fluorescing protein or a red fluorescing protein. And they formed this pattern spontaneously. But if they do the same thing in yeast, so Saccharomyces cerevisiae, we get a different pattern. But it's just a well-mixed set of organisms. So they differentiate into patterns on their own in some way. And those patterns are different. We don't understand this yet, but we need to understand how these organisms are interacting. And then finally, we need to figure out how to get material and information in and out of cells. And this is the bulk of the work that goes on in my lab, actually, is figuring out how material and information are flowing. Primarily, we look right now out of cells. So this is a cartoon of a gram-negative bacteria. So it has a very characteristic shape. These are the flagella that help kind of move it around. The DNA in the middle, and these are the membranes that are blown up here. They're called phospholipid bilayer membranes. So if you remember anything from a early high school biology, that's probably what it would look like in the textbook. And that membrane helps protect the cell, so keeping toxic things out, helps facilitate the crossing in for sugars and other food type substrates, and the efflux, so the transport out of waste products. And it does this all just with a single membrane. What we have at the macro scale is a variety of different membranes and wires and all sorts for all these different molecules. The cell has a single membrane. It does all these jobs. So it's pretty incredible. And we can use, if we could control this, we can use cells much more efficiently to make chemicals, because we need to control what's coming in and what's going out in order to make chemicals more efficiently. We could also potentially control photosynthesis mimics if we can control electron flow into and out of the cells, I mentioned with the bioelectronics example earlier. Or we can even get to detection of toxins in the environment or in the body through sensing by using information flow into and out of the cells. So this idea of moving things across cells is very important, but it turns out that the cartoon that's in a lot of the textbooks is not totally accurate. There's actually a lot of blobs sitting in that membrane, and those are proteins as well. And those proteins are forming pores and transporters for all these different molecules, electrons, and so forth. And so that's what we actually have to engineer. And there's two ways to do that. We can change the transporters that are in there, find the one that does what we want, and put it into a new organism, or we can change the number of them. So if we want to modulate how much is going in and out, we can, let's say if we want to increase it, put more transporters in place. So that's sort of the idea behind a lot of the work that goes on in my lab. What that's led to is we engineered an antibiotic pump to, instead of pumping out antibiotics, which is its natural job for a bacterium that lives in the gut, that's a very sensible transporter to have, something that secretes antibiotics and makes it resistant. But we've actually changed it so that it acts on butanol instead. So we changed the specificity for this transporter so it can pump out the fuel, which is otherwise toxic to it. So if it pumps out the fuel, it can make more of that fuel because it will take a lot longer to reach a toxic level inside the cell. And that actually works when we're trying to produce fuels. That's what this graph shows, is that when we engineer the pump, we get a lot higher titer of butanol. Sorry. Is that my timer?
Yeah, it's your time.
Yeah, okay. That is fine. I have one more example, which I don't have to go through in great detail, but we actually engineered a spider silk to have a, so it's actually not exactly spider silk, it's a different material protein, but very similar, to have an antimicrobial peptide from a frog skin on it, and that material that we made was naturally antimicrobial, so when we didn't have that peptide present, the cells were alive still, they're green, and when we had it present, they were red, so it was killing the salmonella just by being present in our engineered material. So in summary, I think we have to learn to control these complex biological or bio-inspired systems, and there are a lot of ways in which we can do that, but there's a lot of challenges for anyone that wants to tackle them in terms of understanding what's going in and out of cells, but also all of those other challenges I mentioned. So scaffolding for what's going on in cells, learning how they interact with one another, learning how all cells respond to all the inputs that it's receiving, and so forth. So with that, I will keep up a picture of my group. That's my website. If you want to learn a lot more detail about all of the projects going on in my lab, it's at that website. And I can take questions.
Can you just quickly explain how you split the work between, so that there's your group, then there's the Berkeley Synthetic Biology Institute, the Synthetic Biology Engineering Research Center, and I think there's some other place.
Yes.
Why all these different
institutions. So I teach and do research at the University of California, that's always true. But within the university, the collection of us that does synthetic biology research has banded together and formed a Synthetic Biology Institute. So we have some, say, formal but not institution level formality presentations. We get together and talk about our research. Our students can get together and talk about what's going on so that things move faster. And then we have a similar, the Synthetic Biology Engineering Research Center is a multi-institute version of the same sort of society. So we meet twice a year to have all our students talk. And it's MIT, Harvard, UCSF, Stanford, and Berkeley. That's part of that, at least that I know of. And there's probably a couple that I'm missing.
Okay, thanks. Was it? Yeah.
So, I'm just trying to get the terminology correct. And maybe I'm thinking of the Venn diagram. Genetic engineering is it are they intersecting like this, or is one a subset of the other, or so is synthetic biology always genetic engineering? I don't think so, but probably
it's
a
not.
lot of the time it is.
I would say the best explanation is that genetic engineering has turned into synthetic biology. Synthetic biology has become a term that includes much more than that, and there's probably some genetic engineering that is done not for the purpose of engineering biology per se, but for some really alternative different purposes, but most of it is within the synthetic biology umbrella now. Genetic engineering is sort of the level of making changes in the gene.
Mm-hmm.
Synthetic biology is let's make changes that make sense from an engineering perspective. So it's going from tinkering to sort of a mass production level coding.
Is there still an overlap with nanotech? Because I almost thought I almost heard you say nanotech at some point, but I guess not. Maybe not.
You did not hear me say it, but I have spoken on bio-nanotechnology in the past because it does interface quite nicely with synthetic biology. There are a lot of different, so nanotechnology is all about properties of materials being different when you move to the nanoscale level, right? So a nanoparticle of gold doesn't look the same as a molecular gold bar, for instance. And biology is the same way, right? There's molecular level control, and there's differences between the molecular level properties and what we see at the larger scale. And so there's a subset, bio-nanotechnology, where we actually use biology to help create molecules that would have interesting properties at the nanoscale that aren't biological themselves, but we can create them in biology.
But it's not quite to the level where, like Eric Drexler was talking about in engines of creation, where we could potentially use RNA to multiply it many times over, or create software that runs it so it does it by itself.
Definitely not the latter. We could probably do the former. Andrew Belcher has put a lot of effort into using DNA, RNA, and protein synthesis machinery to manufacture nanoscale components. And it's somewhat successful, but I don't think we can scale it up yet for like factory production.
Tammy?
I just have a question about the compartments, because you skipped it, but I'm just wondering what that means, that you're creating new compartments within a cell that do selective things that are different from the normal organelles within the cell?
Yes. So I shouldn't say I'm creating them entirely. Cyanobacteria, so the bacteria that actually do photosynthesis, they house this enzyme called rubisco, and rubisco does carbon fixation for this. So carbon dioxide from the atmosphere being fixed into a sugar in plants is carried out by this enzyme called rubisco. It's also in these cyanobacteria, and it's very poor enzyme. So for something that's so important to us, because this is one of the most abundant enzymes on Earth, it's not very good at converting carbon dioxide into a sugar form, and it acts on oxygen almost as well, and that's a completely wasteful reaction when it does that. And so cyanobacteria actually have these compartments already inside of them. They're made of protein. They look like little viruses, but they aren't viruses. They're very healthy for the cell. And it compartmentalizes this reaction, presumably to help the enzyme do its job better. And so we think we can do the same thing. So we've taken the shell that forms the compartment, we put it in the bacteria that we can work with in the lab, and try to house other reactions that might have a toxic intermediate or a really poor enzyme or something like that.
So my question, so for those who don't know, in Sunnyvale there's an organization called BioCurious, and they are part of this biohacking community, where individual, I mean, just like the people who started the personal computer thing, they were building kits in the garage, right? Something similar is happening in synthetic biology.
Yes.
What's your impression? What's your feeling about this biohacking community? And before you answer, I know Erin Gentry, she spoke at the laser, and we're going to have Raymond and Tito. These are three of the founders, speak at the lasers. So anything you say will be used against you.
Yeah. No, I think in general, the idea of doing that is great because we have really creative people that then have access to something that previously needed a whole lab and at a research institution in order to carry it out. So that's great. I think that we should have some sort of national level set of guidelines for working with the DNA, even for those that are doing it in this way, because the institutions all follow these really detailed guidelines for how to do the work safely. As long as they follow that, I wish there could be many more.
I think they would agree with what you said. But in terms of just feasibility, I mean, in terms of what they can achieve in their garage, which is not really a garage. But
yeah. It takes someone starting in my lab with my guidance, which I have a lot of experience to give to them, about 2 years to get to the point where they can start having ideas on their own and designing their experiments on their own. These are really smart people. They're going to Berkeley. It just takes time to learn everything that's involved. I think somebody can do that in their garage too. It'll probably take a little bit longer without somebody being the manager and telling them exactly what it is they need to look up every step of the way. But it's completely feasible for them after going through that learning curve. It doesn't matter whether you do that learning curve at an institution or in your garage, to come up with some really clever and implement some really clever new ideas. So I somewhat suspect that big question mark of something transformative coming out of this technology will actually come from that kind of space. But it's going to be delayed, right? So we've been working at the research institution level on these problems for a while, and it'll take a little bit of time before they have the knowledge caught up to where we're at. But they've been working on it for some time now too, so we're probably only a couple of years away from that being feasible.
Have you done any work with mycelia?
I have not.
Okay. Is that something less than things?
The thesis of your presentation was something about the nose and how the
I'm
nose.
mentioning those. Yeah. I'd say the thesis of my presentation is that biology is complex and we can learn how to control it. The nose is an example of one aspect of biology where it's pretty complex. It's interpreting a lot of different signals with very few sensors relative to the number of signals in the world.
The other thing, I had read somewhere. Is it true that? And I'm not even necessarily sure what this means. But is it true that I read something like that humans have so many number of genes, and then for example, fruit flies or something have
Yes.
actually more genes? And
Yes.
What does that mean?
That means probably that we don't fully understand the human genome yet, but also to make an organism that has many cells and different functions. So we have a liver, we have eyes, we have a brain, we have all these cells were turned into many different kinds of functions. Fruit flies have all of that too, right? They're smaller, but they still have all these different functions. They have eyes, they have wings, they have all these different functions. And so I think what it tells us is that the level of complexity of a fruit fly is not that different from the level of complexity for us. Our brains are bigger, our heart is stronger, more powerful, but that didn't necessarily require 10 times more genes to encode. It's still, at the complexity level, a similar number of functions for different pieces of the world.
So why would there be more genes?
We don't have wings, for instance.
okay, thank you.
Yeah.